Privileged today to give an update on our COVID-19 Biobank Network Initiative, which really started at the beginning of the pandemic. The initial overarching goal was to create a classical centralized biobank platform with teams of people in various health authorities that allows us to recruit, collect biospecimens, and store them either locally or centrally, but this is all managed with a provincial scope. Obviously, the goal was always to promote access to biospecimens and also enabling COVID-19 research. But very quickly, it became clear to us that that initial perspective required a lot of capital funding, very close involvement with local health authorities who were at the time very busy with pandemic response. So that was not, that initiative initially did not take off as intended. Instead, we kind of switched gears and to become more of a lean structure model, where we became more of a provincial-wide network of partner specimen collection sites. So we basically utilized existing infrastructure, partnered with existing biobanks and public and private labs, again, to maintain our primary initiative objective, which is to collect high-quality biospecimens with relevant clinical data in the COVID-19 research field to be able to support COVID-19 related research. To be more specific, our goals are to build collaborative partnerships with health authorities to really streamline biobanking activities, because classically, researchers have to always struggle to work outside of their health region or a research network to obtain agreements, contracts to allow recruitment and collection of specimens across jurisdiction. So we wanted to overcome that by establishing a partnership with all the public and private labs in BC. We wanted to collect, store biospecimens and associate data from BC patients or participants during the pandemic for secondary research use. We want to develop a transparent, equitable access because of our provincial scope, and a release process of biospecimens and data for researchers who really are, who should be focusing on their research activities, but we want to decrease their administrative burden and ethical concerns, because we want to build a regulatory framework that's approved in our province. We also wanted to develop a biospecimen inventory portal that allow access to provincial COVID-19 biobank samples, and allow additional linkages to clinically relevant information in existing databases across the province, so that the entire process of acquiring specimens and also relevant clinical data could be easily obtained by researchers in BC. So how we want to operate is obviously have stakeholder engagement across the province. We have, in fact, engaged all health authorities and life labs to build a collaborative network to allow the biobank network to use infrastructure that exists in collection sites across the province for research collection of biospecimens, including blood and urine and other type of samples outside of tumor tissues across the province. And with help of local staff, specimens will be processed locally data collection and shipping would occur locally to one of our facilities, where we then categorize and biobank all the necessary biofluids with relevant clinical data in our centrally managed IT platform. And then we would allow specimen and data distribution to relevant researchers if the study is approved by one of our scientific review committees. And again, this process promotes equity, diversity, and inclusion, because we feel that every patient, every participant in BC should have equal access to participate in clinical research, and also researchers across the province should have the ability to access our diverse population, in a sense that we do want to ensure that every geographical area in rural community are also represented in our biobank network, and contribute to biospecimens for research purposes. So here's an overview of our governance model. This took us a while to develop, because we moved away from a classical centralized biobank to a I would call a federated model, where we have a oversight committee, executive steering committee, with representation from academic institutions, from the Ministry of Health, from various health authorities and research institutes. But our core infrastructure is, I would say, first of all, expertise in biobanking. So we have a BC Biobank Network Governance Committee, a provincial operational committee that takes care of a daily operation, a IT platform, which I'll get into a little bit later, that really manages all biospecimen and clinical data in a secure but high quality informatics software. We have a scientific review committee that really reviews in depth the data request applications from either researchers or industry partners. And we also have patient advisory committee that brings us a voice of the patient and participants in the community. Right now, our established BC Biobank Network, or BCBN, sites are Vancouver Island Biobank. It's one of the site that can recruit patient, also process and provide storage capacity to BCBN. We have Providence Health, particularly the post-COVID recovery clinic, and the CANCOF study provides recruitment of both inpatient and outpatient population, and specimen collection and some of the storage capacity at locally at St. Paul's Hospital. BC Children's Hospital Biobank is a very well-established biobank in our province, and it is one of our partner sites that does active recruitment, specimen collection processing, and also to a certain extent storage as well. Lastly, Vancouver General Hospital is also set up with a post-COVID recovery clinic, which recruits COVID-19 patients and also processes specimen that they collect. We have a working group that is represented by every health authority, and all of us in that working group would like to contribute to provincial biobanking activities. However, right now, Fraser Health is still in development, because they have a different model where they want to contribute with a research coordinator instead of us using their outpatient collection sites due to capacity issues. Interior Health is also struggling with staffing their labs with phlebotomists, so they're a little bit behind, but they are on board with the model and the objective. However, it's just operationalizing it in their health region has becoming a little bit of a challenge. Of course, Northern Health, we are also engaged with them. Hopefully in the future, we would be able to expand our footprint in those health regions. That being said, we do have agreement with Life Labs that allows us to recruit and collect patients across the province. So in our governance model, it states that every partner site within the Biobank network can participate as part of the governance committee, which allows them to provide leadership, oversight, and direction for activities within the network. So they do have, as a partner site, they do have a say in how the network functions. They also participate in the scientific review committee to review requests and also facilitate release of biospecimen in an equitable fashion, including, of course, relevant clinical data. I think most importantly, what makes this operation successful was that we allowed partner sites to remain in control of their own biobanking operations, including specimens that they have processed, so that this is really a collaborative effort where, in a sense, we're a true network of biobanks rather than one single team that controls all the samples and its operation, but we rather would rely on the federated model to ensure our continuous success. Partner sites can contribute if they have capacity, and they can also utilize our existing infrastructure, resources, and equipment as need. So there's a mutual benefit to be part of our network in a sense that we are able to provide some infrastructure and resources if such things are required. So not only do we have our own protocol to recruit and collect patient with COVID-19 infection, we also partner with other PIs and studies to support their research from a biobanking perspective. I think the best example is the post-COVID recovery clinic, which in fact has a presence, I think, across the province. We as a network will develop partnerships with research entities or our PIs across the province that require access to our infrastructure. In this particular case, we in fact oversee now the entire biobanking operation for the post-COVID recovery clinics across the province. And they use our consent form, our collection process, SOPs. We do provide storage of data and specimen. And in terms of distribution of biospecimens, the PCRC works on our access committee to review applications and grant access if it deemed reasonable. We benefit from this partnership because PCRC and other research studies, they have often a very well established mechanism for referring patient. Sometimes they have good relationship with communities where we would leverage that to encourage and promote participation and biobanking. So again, there are mutual benefits to be had as each partners or researchers could focus on their research activities, and we would really take over the biobanking activities so that there's really lack of duplication of work and infrastructure. So here are our recruitment strategies. We do actively recruit patients, again, through the PCRCs. We have recruiters at BC Children's Hospital Biobank, and also we recruit through the BCCDC COVID-19 consent registry. CANCOV is a study that recruits inpatient with COVID-19 infection at Providence Health. So we do reach out to patient and recruit and consent them. But we also rely heavily on passive recruitment. And this is through our e-consent platform, which allows anybody in BC to look at our study, to participate and donate biospecimens. And we do that through our website and also through the Reach BC platform, which is another very well established portal. So in terms of participant enrollment, again, because of the COVID-19 pandemic, dealing with paper-based consent and signatures in a way that's safe for a participant and for our recruiters can be quite challenging. So we now has switched to the e-consent form, which allows the participant to use either a computer or a mobile device to fill in the necessary information. There's a short questionnaire that really asks them about their COVID-19 vaccination status and infection status, which will then automatically be stored in a REDCap database, and which is also linked to our Open Specimen Biobank IT platform. So information regarding patient demographics, clinically relevant information are digitized automatically and stored with a sample or patient identifier. Then a requisition will be generated electronically, and this requisition is recognized at all lab locations across the province, whether whether the patient would present at the Life Labs or a hospital lab where applicable. This is because not all hospital labs has capacity to provide the service. So there is some coordination that is involved, but overall, between what Life Labs could offer and our partner sites in various health authorities, we are able to capture most of the population in our province. We are still working on Interior Health Region and Northern Health Region. But as you could see, that the enrollment has been rather streamlined in an electronic fashion. So what are the patient consenting to? Again, we're talking about our own BC Biobank network study. We do support other research studies that might have a different protocol. So we do provide fit-for-purpose biobanking needs. But in our main study, we are consenting patient to donate one serum tube, two EDTA plasma tubes, and also one more EDTA tube for whole blood. This is for DNA extraction purposes. There has been a lot of discussion on whether we should include PBMCs or extract puffy codes for additional research studies. Not all collection sites are able to accommodate that. Some of them don't have the right equipment or expertise to do additional processing. So we then decided to go with more of a basic collection protocol where this is more feasible across all collection sites in the province. Again, we do collect patient demographic information through the e-consent and also relevant COVID history in our questionnaire, e-questionnaire. There is an option for patients or participants to consent to have their personal data linked to other databases in our province, especially the Pope Data BC platform. And they also have the option to consent to be informed about any incidental findings that might occur. I want to quickly introduce the Open Specimen platform, which is a provincial biobanking platform that we have set up in collaboration and support with the UBC Faculty of Medicine Digital Solution Team. I think they used to be called MedIT team. This is something that it's now centrally managed and supported and maintained by a Faculty of Medicine Digital Solution Team. It is compliant with all the UBC security and technical standards. But most importantly, it's quite easy to use because you can access this platform or the software using your UBC campus-wide logins. So potentially any anybody with the UBCY login can access and view their inventory at their workstation. The utility of or the advantage of using this open specimen platform, first of all, it's one of the most common platform that biobanks use. So there's great compatibility across various biobanks that currently exist in our province. But ultimately, it provides the ability to track specimens from collection to release and utilization. It also collects annotated clinical data based on the project's needs, but it has the ability not just to link with other databases, but also host itself clinical data with the right biospecimens. And if the clinical data is complex, the Biobank, the open specimen platform can integrate with other data platforms such as REDCap, EPIC, or other lab information systems that we use. Currently, BCCBN or BC COVID-19 Biobank Network exclusively uses this platform. We have onboarded over 2,000 specimens. I think we might be at double that figure now. There is a quote-unquote data entry process when we receive the specimens, even though they are trackable to each collection site, but we do have to generate additional barcode labels and enter it into the open specimen or scan it into the open specimen system. So there is some data entry that are involved, which requires hands-on time from our staff. I think this platform is very powerful, and certainly Providence Health and some of the other biobanks are either using this or they are actually using our platform to support their biobanking needs. So again, we are having a lot of discussions with other academic centers or biobanks to leverage this provincial platform so that we could promote some crosstalk between biobanks in terms of their to get a better understanding of how the inventory systems, how the inventory of biospecimens landscape look like in the province, and provide easier access to researchers when they're looking for existing biospecimens for research purposes. So here's our pathway to obtain biospecimens within the Biobank network. First of all, there is a intake form that is centralized to our website, and the BCCBN is a part of, started as a working group of the COVID-19 Clinical Research Coordination Initiative at UBC. So we still use their website, but we are working on our own website. But there is a application form that needs to be submitted on the web, after which we will provide feedback to the applicant with an estimated of sample size and volume of biospecimen that is available to the researcher, and also a cost for in a cost recovery under a cost recovery model. Then the BC COVID-19 Biobank Network Scientific Review Committee will assess the application, and if approved, then there might be additional supporting documents that needs to be placed, including material transfer agreement, and ultimately specimens and data are then released from our Biobank Network sites. So specimens that are collected and stored at Island Health can be then released to researchers in Fraser Health, for example, because we're all operating under the same governance structure. So here's an overview of our inventory that comes from the post-COVID recovery clinic. We have to date over 870 participants. So these are recruited through the PCRC, over 4,300 primary specimens and over 47,000 aliquots. Some of these are longitudinally collected, which are very useful for longitudinal studies. But as you can see the breakdown, most of them are plasma serum samples. Some of them are PBMC. We also have some puffy coats and RNA packaging tubes. Again, the distribution here is due to the ability of various PCRC clinics to process collected specimens. So certain sites are better equipped to process PBMCs or puffy coat, isolate puffy coat layers. Most sites do not have that ability to do so. Our BCBN study, in fact, we had a lot of challenges at the beginning to obtain REB approval, partly because of the nature of the study, which is provincial scope, has a lot of players, many stakeholders, and also a study that is not focused on any particular condition other than a patient needs to have either a COVID-19 infection or received COVID-19 vaccination. So there was a lot of back and forth with REB and also various health regions, but ultimately we were able to launch our own study earlier, I think, end of last year. And this is all through passive recruitment through our website or the ReachBC platform. So far, we have 87 participants. Now, these are participants who have consented. Not all of them have present themselves to a collection site yet. So far, we only collected 89 primary specimens. And over 800 aliquots. So we're still waiting for some of them to come to one of our collection sites, but we kind of have a process of reaching out to participants in our province in a very low cost but high yield fashion. So as a side that we also support PI-driven studies because we want to focus on COVID-19 research, but at the same time, biobanking is biobanking. So we don't necessarily need to restrict our efforts to COVID-19 research. There are other studies in the province that reached out to us in terms of supporting them with biobanking needs. So I think the biggest one is the CanSolve study, which is a CKD vaccine study led by Dr. Adira Levin. So we provided recruitment, collection, and processing and storage of all the hemodialysis patients that consented to the study across the province. And according to the protocol, all of the biospecimen were to be sent to U of T because that's where their central biobank is located. But we were able to, again, because we provided all the biobank work, we were able to generate using remnant samples, our own inventory of over 644 specimens longitudinally collected over 5 visits, and each of them have 4 aliquots. That's after we have met our deliverable to the U of T biobank, and we have this cohort locally for secondary research use. We also have supported a menstruation ovulation study led by Dr. Geraldine Pryor, where we do have over 200 saliva samples. another endometrial cancer study in collaboration with BC Cancer. I think the PI is Dr. Andrea Nelson. We now have a mix of serum and plasma samples from endometrial cancer patients. Some legacy specimens from, I believe this was Vancouver Coastal, where remnant specimens from COVID-19 positive patient in the ICU was processed and stored. So we have over 300 of those plasma samples within our biobank as well. So not all of these samples are ready for secondary use without any restriction. Obviously, PI-led studies have REBs that might or might not allow your type of the research that you're interested in. So typically when we receive a request to access, we have to review both what is being, what researchers requesting them, and also what the REB allows us to do. But let's just demonstrate that we can support research in our province outside of COVID-19, and we intend to do that. And also that having the ability to collect across a province and process and store locally is a very cost-effective way of doing biobanks. I think I would like to provide a summary of our developments and also describe their benefits. So some of the things we developed, which I discussed prior to this slide, but I just want to stress the fact that the electronic consent model it's very user-friendly. It's low cost to maintain, certainly a lot cheaper than having to actively recruit patients. And it's very welcomed in remote communities. So this allows us really to generate a large representation of our patient population or healthy population in our province. Once consented, the collection protocol and the consent form is actually electronically sent to life labs across BC. So participants or patients don't necessarily have to bring the right paperwork with them to the collection site. That often is a problem when they brought the protocol but not the consent form, or the other way around, collection could not take place. If they physically show up with their care card at one of the life labs collection site, life labs is able to pull out relevant information directly on their system, so that this allows easier access and allows us to really increase our throughput in recruitment. That this function is not yet available through the public labs due to technical difficulties, but I think we will get there once we have a better automated software. Because we have a provincial scope, we coordinated extensively with provincial laboratories, again, in partnership and in support by the Provincial Laboratory Medicine Services. We have a working group that has representation from each health authority in our province, operations and sometimes medical leadership. And the goal here is to really reduce the burden on researchers and public labs with contracts and agreements that needs to occur before research can happen or before collection could occur. So we hope to have, we do right now have our requisition recognized by all provincial laboratories. We are working on a single cost model and also material transfer agreements across the province. So that really hopefully would expedite research and decrease burdens on PIs to figure those things out. We again developed a centralized IT platform supported by UBC. So that is something we want to promote because it really moves away from us working in silos, but rather have a centrally managed solution that could meet our needs, but also allows us to be more transparent with and equitable when we want to support research. Our provincial-wide collection of biospecimens are held to a very high standard, and we are actually part of a national biobank network, which requires us to meet certain quality standards. And this we are actually also working on a national alliance on biobanking initiative, which would really give us greater visibility when we need to apply for funding and collaborate in research areas. So I think the take-home message for people who are doing research is that we do provide access to coordinated and high-quality biospecimens. We can provide linkage to data sets that annotate biospecimens either when we collect them for you, or provide linkages to existing databases in our province if consent allows us. We do provide fit-for-purpose biobanking needs for specialized services, and some of our genetics study partners, for example, a whole-seq program required very specialized processing and storage. So we could provide that type of service, although it might be restricted to certain sites because it's right now it's not realistic to expect all collection sites across the province to meet your needs because because of lack of expertise and equipment. If you want to achieve your biobanking needs through the BCCBN, we obviously will set you up with the account to manage your own inventory through the open specimen platform. So that's another value add that we do provide. So future directions, I hope as we move past COVID-19 pandemic, we are hoping to expand our scope to support clinical research in all areas, because biobanking doesn't discriminate disease conditions. It really requires us to build inventory that is not focusing on plasma serum, but also other specimen types, so that we could have a central portal for specimen release that meets everyone's needs. So I think the only thing that out of scope is tumor specimens. We don't have the capacity and expertise to tackle that area. But in terms of biofluid, we do want to expand our focus and our specimen types. We want to continue to collaborate with our health authorities. We don't have material transfer agreements with every health authority, and some of the costing model might require some additional tweaking. But the goal is obviously to have a streamlined process so that when you come to the biobank network, you don't have to worry about the paperwork, the agreements. We will provide you with a cost for your particular study, and you can focus on the research. We want to continue to develop and expand the provincial IT platform because it does reduce administrative burden for all of us. It is very cost effective and efficient, and again, it promotes collaboration and crosstalk between researchers and biobanks. We want to develop a provincial biospecimen catalog using the same open specimen platform, because it can be a web-based portal that allows internal and external access to both academics and industry partners. so that we have a good idea of who is, which PI is doing which study and what type of specimens are available in a central location to better enable access to biospecimens and research. Again, we will continue our national participation in the Data Alliance and Standardization program, so that the quality of our biospecimen and clinical data are up to standards. Finally, we are very gracious to our founders, UBC Faculty of Medicine, Genome BC. We also received funding from donors from the St. Paul's Hospital Foundation, and I think also the UBC Faculty of Medicine program. And here are our partners. And if you want to contact us, you can either contact me directly or reach out to one of our staff. Our temporary website still is embedded within the CRCI MedUBC portal, but we will be posting a new website which allows just easier access. Thank you very much.
Thank you so much, Michael, for that presentation. There's a comment